Assalamualaikum and very good morning to Prof. Dr. Wan Kilan and my fellow friends. The title of this article is Complete Chloroplast Genome Sequence of the Medicinal Plant Aconitum transectum raniculaceae Comparative Analysis and Phylogenetic Relationship that has been published on 2023 by Springer Nature. Before I continue the presentation, I would like to introduce myself and also my group member. My name is Normiza Ismahani binti Khairul Nizam and my group member is Howdy Shen, Lee Yilin and also Amira Nabihah Najwa. We would like to present this article based on 4 sub-content, which is introductions, methodology, result and discussion and also comment and conclusion. Aconitum transectum is a medicinal plant that used widely in the traditional Chinese medicine. This species in the family of Ranunculaceae, genus Aconitum, and has 3 subgenera, which are Aconitum, Gymnaconitum, and also Paraconitum. In this article, we will be specially discuss about the species of Aconitum transectum. Its morphological trait make it difficult to differentiate the Aconitum transectum with the other species of Aconitum. It is very important because using the wrong species of Aconitum will potentially endanger the patient life. Even though this species make a great contribution in the Chinese medicine, but there is no research has sequenced the chloroplast genome of Aconitum transectum. In this article, we would like to highlight about the chloroplast. Chloroplast whole genome data analysis will become an efficient tool for species identification and species evolution studies. Thus, we have to know what is chloroplast. Chloroplast is an organelle that can be found in the green plant and algae. It is a location of the photosynthesis in plants, and it has own genetic system consisting of closed loop of double-stranded DNA molecule and also capable of semi-autonomous replication. The structure of the chloroplast is tetramic structure with a large single copy, a small single copy, and also have 2 inverted repeat, which are IRA and also IRB. And the chloroplast is small and highly conserved genome structure. Next, the objective of this article study is to identify and compare the difference between the chloroplast genome of Aconitum transectum and other related species, to conduct a phylogenetic analysis using the chloroplast genomic data, and also to provide the theoretical foundation for understanding Aconitum transectum's chloroplast genomic characteristic and phylogenetic relationship.
Okay, let us move on to the methodology part of this article. First, the researcher harvests Aconitum transectum fresh young leaf from Machang village, Lijiang City, China. Then they submit this fresh young leaf sample to company for sequencing purposes. Then they do whole genomic DNA extraction from this fresh young leaf sample by using the cetyl-trimethyl ammonium bromide method, which it can remove polysaccharide contaminants from the plant DNA. Okay, move on to paired end sequencing by using Illumina NovaSeq 6000 platform with 150 sequencing read lengths. Next, they screen the raw data to get high-quality, clean data by using FastP software. They assembled the chloroplast genome by using SPECS with k-means value of 555, 87, and 121. Then they do the quality control of the chloroplast genome assembly by using sequence of Aconitum purpurans with this accession number below. They use 3 different software to annotate for 3 different targets, which Prodigal is used to annotate for the chloroplast coding sequence, CDS. Then HMMER is used to predict the rRNA, and Aragon is used to predict tRNA. Then to produce the second annotation, they check the assembled sequence using BLAST. Then they produce second annotation based on sequence of related species they found in gene bank. Then for final annotation, they manually verify 2 annotation results to remove incorrect or redundant annotation and to establish exon boundaries. Last, they map the genome by using OGDraw software. Next, they perform codon usage and repeat sequence analysis, which since the genomic codon usage rate differs widely between species to species and organisms to organisms, then they calculate these RSEU for each codon by using the codon W software. This inequality in codon usage is due to a combination of natural selection, species mutation, and also genetic drift. Next, move on to comparison of complete chloroplast genome. They compare between chloroplast genome of Aconitum transectum and 6 other reported Aconitum species and 1 exogenous species as listed as below. By using 3 different software. The CGView software is used to evaluate chloroplast genome structures. The mouse software is used to analyze homology and covariance of chloroplast genome. And then the MAFFT automatic move software is used for broad comparison of homologous sequence from different plants. Next, they calculate the nucleotide diversity value for each gene using DNASP software. Then they visualize the inverted repeats, small single copy, large single copy region boundary information to compare between gene sequence by using SVG package. This Ka is the non-synonymous substitution rate, and Ks is synonymous substitution rate. They calculate the Ka to Ks ratio value of gene by using KAKS calculator software. Lastly, the researcher performed phylogenetic evaluation where they obtained additional 27 chloroplast genome sequence and YCF1 gene sequence from the gene bank as shown in the right side of the slide. And then they conduct sequence alignment based on chloroplast genome of these 28 species by using MAFFT Trim AI software. Then they construct maximum likelihood phylogenetic tree by using IQ-tree software, where they use the Delphinium unanis and Delphinium brunonianum as outgroups and bootstrap value of 1000. And then they select the best tree building module by using built-in model finder of IQ-tree.
We will now be moving on to the results obtained during this study. The chloroplast genome characterization, as you can see in the table, was obtained by the sequencing, assembling, and annotating of chloroplast genome of A transectum. The words CP you see in the table refers to chloroplast. As highlighted in yellow, the A transectum CP genome is 155,872 base pairs tetrametric structure, including the ones highlighted in orange, which is large single copy LSC with the base pairs of 87,671, and small single copy SSC with the base pairs of 18,891. Also included in the genome structure are the ones highlighted in green, which is a pair of inverted repeat sequences IRA and IRB with 25,894 base pairs each. As highlighted in blue, you can see that the complete CP genes have 131 genes that are encoded, with the comprising of 86 protein coding genes, 37 tRNAs, and 8 RNAs. Next is the codon usage bias, also known as CUB, which we used. CUB is a concept that describes the differential frequency with numerous synonymous codons encoding the same amino acids. As seen in the table, we obtained the codon list of A transectum by examining the codon usage frequency of protein coding genes in the A transectum CP genome. What we found was that the most codons were leucine with 2,243 codons, cysteine, which was the least codons with 308, and we had the most favorite codon, which was AUG, which encodes for methionine and had an RSC value of 6.9783. Next is the interspersed repeats, where we discovered 46 of them in the A transectum CP genome. As you can see in the graph, I divided the graph into 3 for easier understanding. In the first half, we discovered 35 repeats with the length between 30 to 39 base pairs. In the second half, we discovered 10 repeats with the length of 42 to 107 base pairs, and in the third half, we discovered 1 repeat with the length of 26,229 base pairs. Moving on to the SSRs, we identified 241 SSRs, where 137 of them were discovered in the intergenic spaces and 104 were discovered in genes such as YCF3, YCF2, YCF1, and many more. The A transectum CP genome is similar in size, gene composition, IR expansion, and contraction to the CP genomes of 7 Renunciales species. The phylogenetic analysis of CP genomes of 28 plants from the Renunciales family shows that A transectum is most closely related to the ones circled in red, which is A. vilmonorianum, A. escopal, and A. foresti of subgem Aconitum.
The CP genome of A transectum, like most land plant CP genome, has a highly conserved structure and gene content. So the genome has typical tetrastructure with 4 segments, LSC, SSC, and 2 IR, and highly conserved IR regions, and is native to northwestern Yunnan, and its CP genome size is similar to the other Aconitum species. The SSC region is longer compared to other species. Nucleotide diversity (PI) indicate that the response to the degree of variation in DNA sequence, and nucleotide diversity respond to the genetic diversity of the species. The higher PI value of gene sequences in the LSC SSC region were observed from the IR region, which is coherent with other endosperm. RPS19 gene has a tendency to enter the IR region due to the expansion of the LSC IR boundary in the genome aconitum, while the Yunanes, the LSC IR boundary is continental and the RPS19 gene are placed in the LSC region. It indicates that the CP genome boundary gene of the genome aconitum are different from the Yunanes and those other genera in the Ranunculaceae family. In this study, we found that A transectum has 46 repetitive sequences, which is much higher than that of other species in genus Aconitum. Furthermore, the majority of the repeats are found in the gene, indicate that A transectum CP genome preserves a lot of genetic information. SSR has been found frequently utilized to determine phylogenetic relationship, genetic diversity research, and species identification due to their high variability and recessive inheritance. It contains 241 SSR, the majority of which were identified in the LSC region. The YCF1 gene, the largest chloroplast gene, encountered an ATP binding SSABC protein in chloroplast and generally evolved at a rapid mutation rate as formalized in our study. YCF1 is the most potential chloroplast DNA backup for land plants. It is very species-specific. The YCF1 polygenetic analysis also showed that YCF1 has taxonomy potential at the subgenome level within the genus Aconitum. The KAKS of YCF1 gene were larger than 1, implying that YCF1 gene were positive selected to adapt to the living environment. Based on morphological feature, nuclear DNA marker, and some CP genome are concrete with our phylogenetic conclusion. Based on ITS sequences, a phylogenetic tree for 51 species of the genus Aconitum, 27 CP genome of Aconitum species previously created. The phylogenetic of this ITS sequence show that A etc., A Vimoridanium, A Episcopia, and A Forestier are in the same clade and belong to the same subgene, Aconitum.
Last but not least, what we can comment and conclude about this article study is first, the complete chloroplast genome provide a crucial reference for Aconitum transectum. The chloroplast genome of Aconitum transectum and other Aconitum may be similar in some term, but it will be differ in the inverted repeat region boundary section. Second, phylogenetic races of Aconitum. The chloroplast DNA is a barcode for the green and land plants. It will give the informatic marker for phylogenetic races of Aconitum. From this, the close relationship can be discovered between the Aconitum transectum and other related species of Aconitum. And third, it will be source of the reference data that will be used for further study of the population genomic, phylogenetical analysis, and also genetic engineering research. That's all from us. Thank you for lending me your ears.